Perfect.
You just need to use this thing.
I need to use this thing.
Steering it.
So big data may not be a challenge, but this certainly is, and that's the, okay. So thank you very much to the President, to the Chancellor. It's a great honor for this Irish Catholic to be here. And today I'm going to talk about what might be a fairly uncontroversial aspect of big data, but I will highlight some areas where perhaps we need to think. I was struck in the middle of the night that Rome was the origin of the first big data. Caesar Augustus asked a census of all the people in the Roman Empire, which extended all the way to quite close to Kaust, where we live in Jeddah, to a small family, a man, Joseph and Mary, and she was pregnant. This is one aspect of big data perhaps people don't think about. So today I'm talking about a phylogenomic investigation into the origins of cyanobacteria, which is part, of course, the biosphere. When you consider microbes, they are frequently underestimated in the activities they carry out. They are no less than fundamental to our entire life on the planet. Just as an example, half of the CO2 that is cycled by microbes in the marine environment. It underpins ecology on the terrestrial environment, human health and disease. The microbiome studies indicate that. Our understanding of microbes is incredibly poor. If you regard this small table, species, and we could argue the species concept, but certainly if you accept it at the first approximation, you have between 10 to the 5 and 10 to the 7 species that have been described. And the defined species, as in those that have been cultured, is merely 12,000. Now, we have 17,420 genomes as of a few months ago. Those represent pangenomes as well as species genomes. Archaea, of course, a smaller group, and only 362 have been sequenced. Sequencing is coming into its own here because of the changes in technology. But what can we do with it? If you look at the output of sequencing, you can see that it is near exponential. However, the coverage is very, very focused, mostly on 6 groups. There are presently 47,000 genome sequence projects. This does not include the metagenomic data. So these are targeting specific organisms. Thinking in terms of strain collections, there's about 724 plus or minus strain collections in the world, and between them they hold about 1 million strains. So we are orders of magnitude underestimating or undermeasuring the diversity of this group of organisms. What can you do with it? I think a genome provides a genetic model, and from that model you can generate metabolic models. You can also understand capabilities at population level. You can make predictions based on these data. It is a systematic and intellectual framework within which you can place experimental data in a well-understood system and extrapolate to a less well-understood system. In this particular picture you have on the left Syneca cystis, which is a model microbial cell factory, which in theory would facilitate production of molecules without generating a CO2 footprint. And on the right you see Mycobacterium tuberculosis, something I did at Cambridge, and there is looking at evolution of the organism in a extreme drug-resistant set of strains. So you can use this technology for good. You can also use it for bad. In this talk, I'm really going to talk about the phylogenetic analyses. And here's Klimt's L'Arbre de Vie, a beautiful, beautiful picture, 1909, way before any of us really thought about phylogenomics. But it's absolutely fantastic. The tree of life is really a mechanism for placing experimental data in context. So what I was want to do here is use big data to do just that with the cyanobacteria. For those of you who have yet to meet cyanobacteria, and they are wonderful, they're gram-negatives, basically E. coli-like. They are the origin of oxygenic photosynthesis. They're very ancient. They evolved under a time when there was no oxygen. The UV they can withstand is absolutely huge because there was no oxygen to produce an ozone layer. They're widespread in nature. There are even some found in dark caves, though they themselves have lost the capability to be photosynthetic. They are critical in the marine ecosystem where they are the primary producers. Oh, I should say these structures are a taxonomic approach, and as you can see, as you go from 1 to 5, there is an elaboration in physical form. Section 1 is a binary dividing cell. Section 2, you have a second plane of division, just visible in this one. Section 3, it's multicellular, but the cells are all equivalent. Section 4, it's multicellular with some level of differentiation into reproductive cells, into nitrogen-fixing cells. And number 5, they're the cathedrals of cyanobacteria. In those, they invented a right-hand turn, and so you can actually produce scaffolds. This simple set of structures are largely unchanged across the millennia. There are stone fossils that reveal very, very similar structures, if not indistinguishable from today. When you consider the marine cyanobacteria, and this is satellite imagery, we're blessed today to have a satellite specialist, so I'm somewhat scared, but I understand this is done by spectral analysis. What we're looking at here is the placement and frequency of particular spectral emissions that come from marine cyanobacterial pigments. There are two groups being looked at here. There's the Synechococcus, which in the graph, which is poorly placed, they tend to occur in the first 50 meters of the sea. They are a high light, high temperature group of organisms. The far more numerous, and in ecological terms far more important Prochlorococcus, will extend down into 200-meter depths, depending on the turbidity of the water. And they are the main workhorses of the primary production. The abundance, as you can see, the Prochlorococcus are concentrated in the warm, high light areas, makes sense really, whilst the Synechococcus far more broad spread because they're at the top of the surface, and that is an easier place for them to live. If you think about phylogeny, and everybody knows about 16S phylogeny, it is a very, very effective tool. And given the technology of the time when it was invented, there's nothing to beat it in a way. Simple PCR followed by a sequence, and then you can derive some level of coordination with the known taxonomy. That has elaborated over time by the inclusion, because the 16S is a single gene and it is also subject to gene flow, you can have 16S genes moving in cyanobacteria. So this would be very confusing taxonomically. So people add more genes, but the genes they're adding typically are involved in central architectural components of the cell that do not evolve particularly quickly, obviously, and they themselves have limitations. One of the big limitations is that you concatenate the whole sequence and then start removing data in order to allow a clear comparison. So you are cutting the data down. Nevertheless, a relatively robust phylogeny has been developed, but it is not taking in all the data. When we look at what a microbe is, it is the sum total of its genome. That includes plasmids. If you think about diphtheria, Corynebacterium diphtheria is not a pathogen unless it has the diptox plasmid, in which case it is. If you think about other cells, Streptomyces, for instance, those large linear plasmids carrying huge biosynthetic pathways, again, completely change the biology of the cell. The cell is the sum of its genome. Our single gene or even multi-gene phylogenies don't address that. They merely look at what is the history of those genes they look at. They don't address the pangenome. Pangenome refers to the fact that a species can have a core genome, and then there seems to be additional genes coming in and coming out as a result of gene transfer. The pangenome varies. Mycobacterium tuberculosis does not engage in horizontal gene trove, not in the last few millennia, and it is effectively a very tightly prescribed species. Others that engage in a lot of gene transfer, typically living in a highly variable environment, will have genomes that vary tremendously between members of the same species. When we look at cyanobacteria, there is a significant problem if you ignore the hypothetical proteins, many of which are unique to the cyanobacteria but highly maintained. All these are is important genes for which we have no function at the time. And these are, ab initio, removed from such single gene or multi-gene systems. Finally, we have got an RNA level of control. Increasingly, RNA control is coming in as a major component of gene regulation. And that is encoded directly in the DNA, but again, we miss it. So if we were to use big data, we could attempt to address these difficulties. In the department, we have made a program, this is work between Vlad and myself, in which we use a very high-end computer to carry out if you will, an in silico DNA hybridization. As a microbiologist, the gold standard of speciation is in fact a DNA hybridization done on hydroxyapatite. It's a very simple technique, and you can do it against a panel and rapidly generate understanding. We do the same thing now using a computer. The computer carries out a reciprocal genome comparison. It computes the genetic distance, and then from that derives a tree. And you can see there's a small example here. So to really look at the cyanobacterial genome, there are presently over 180 cyanobacterial genomes extant. These are all from defined strains growing. This is not counting the uncultured metagenomic cyanobacterial genome. And in KAUST, we've generated an additional 10% to this group here in the last few months. So this is an expanding area of investigation. I've taken this rather busy slide. I do apologize, and we'll just blip through it. At the bottom, you can see the 5 sections, which are the taxonomic delineations within the cyanobacteria, and they are coded blue, green, yellow, orange, and red. And as you can see, the size of the genome does, to an extent, relate to the complexity of the cell structure. So this would suggest, gosh, you need a big genome to be multicellular. Well, in the interests of literacy, we'll leave that one hanging until the end. What you can see is the genomes vary from incredibly small, the picocyanobacteria. These are organisms, the Prochlorococcus, Synechococcus. Their energy budgets are incredibly constrained. The genome is minimal. You're looking at 1.6 megabase genome. I mean, it's very close to not having enough genes to function. These organisms are the ones that are driving most of the carbon flux in the cells, in the sea. And this goes all the way up to members of the 4 and 5, where they tend to be terrestrial. Okay? As you come from the left to the right, you become increasingly terrestrial. Okay? There are marine species at this end, of course, but the marine environment is selecting for small, highly efficient cells. Once you hit land, you can start to be less specialized and become larger. I'm anxious of time, so we'll just blip to this. This is a challenge of big data. There are only 178 genomes on this, and none of us can read any of them. This is one of the aspects that human beings are very poor at understanding this. The journal is happy because I said everyone looks at this as a PDF and they'll blow it up to see the various points. But if I could just sort of expand, that does work. Here are the Cyanobacteria. These are the marine organisms, okay? Now, what's happened is we have got a very early differentiation between this group and the taxonomically broader and the structurally far more variable terrestrial and some marine strains here. Where there's a red dot, you have got a switch between structure. A filamentous becomes unicellular, or a unicellular becomes filamentous. Filamentous becomes ever more specialized. This group here, which you can't really see, I do apologize. This group here is the 4-5 group. So in other words, they evolved relatively late. It's believed something like 1.8 billion years ago. Somebody invented nitrogen fixation. Nitrogenase cannot operate in the presence of oxygen. It destroys the enzyme. So you start to specialize cells that carry out nitrogen fixation and keep them away from oxygen-generating photosynthesis. This was highly successful, and this group, the 4s and 5s, are very well spread across the world. In this area, they don't fix nitrogen. These organisms rely on someone else doing it. Now, while we are looking at this group, you can see these red lines. I'll get back to that in a second, but just be aware that we seem to have some structure and some organization. Where there's a gray dot, there is limited evidence for that bifurcation in the phylogenetic map. If we take this group here at the top, oh, sorry, this one. I've just blown up. We're looking at unicellular, subsection 1, marine Cyanobacteria. When you look at them, they form basically 3 clades, 3 subclades, and this highlight 2, highlight 1, low light 4, 3, low light 1, and low light 4, these are assignments made on the basis of the ITS sequence between the 16S and the 23S, on the basis of a mere 100 and something nucleotides, okay? And this is an ecotype. And what you're seeing is specialization of a cell to a particular depth, salinity, and light condition. And it was assessed by a very short piece of the genome. Our map is made by comparing the entire genome, 2+ megabases. And what you see is we're reproducing the ITS-based system. That is evidence, I would say, for a genome-wide adaptation. These genes are co-evolved, and you can actually detect that signal still in the genome. So despite this being big data, it does exhibit extreme fine differentiation. So coming close to a summary, so that we have plenty of time for discussion, our evidence is that the Cyanobacteria diverged very early from the rest of the cyanobacterial groups. Light-adapted Cyanobacteria have a genome-wide co-adaptation, which was first picked up with the ITS sequence, but now we see it reproduced. Structurally diverse 2, 3, 4, and 5 are likely to have arisen from unicellular cells. That's our conclusion. Others previously on 16S placed it as filamentous, then when it became unicellular. We don't see the same evidence in the whole genome. One thing that I mentioned was the larger you get, the more likely you'll have a higher level of structural diversity. Our evidence is that you do not need a large number of genes to change from unicellular to multicellular. And given the very long presence in the biosphere of these organisms, one is proposing that, in fact, the structural determination is the result of a few genes being transferred across a set of genomes that have a capability to support multiple structures. Thinking about that, and just as a suggestion of how difficult that may be, for a multicellular, for the filamentous organism, it's a gram-negative. The outer membrane is shared between cells. So it is an interesting biological adaptation. And we see very clear evidence that the 4 and the 5 have a monophyletic origin and appear descendant from Trichodesmon, which is sometimes known as marine dust. You see it's sawdust on the sea. And with that, I'd like to thank my group. I'd like to thank Vladimir. And was that the time? Khalas. Thank you so much.